If you have any questions, you guys can pick these up at the end of class, or however you want to do that.
Yep, that works perfectly. And I just have to brag a little bit on these ladies because this was their poster presentation when they took the class. So they did a microgaining poster. From that, I looked at the work that they had done, which was very pedagogically rigorous. They did an excellent job. And I said, we should do this. We should launch this. And we did for real last semester, incredibly, as Christy mentioned, very wildly successful. But not just that, not just did students like it, they also gleaned a lot of very useful information. And these ladies did an iterative assessment of what had been successful in the gaming option. And there were incredible reflective practitioners to the point where this semester we'll be presenting at an educational forum, presenting to other faculty about this idea of integrating games into teaching. So how rad is that, right? So that's where your poster may lead you, or to something similar. So thank you for coming. We really appreciate having you here. And think about that as a possible option. A lot of you are pondering right now because choices are hard. I've given you 3 super appealing choices. And I can't tell you right now, I don't have good data to tell you which one is going to be the best one. But here's the thing. I think that's very individual. And I personally believe that if you are stoked about the option you choose, it will inspire you to learn, and that will relate to your performance. So just go with the one that interests you the most. Don't try to think about what is going to make me more successful in Rachel's class. If you are happy and you are learning, you will be successful. Okay? Thank you. All right. We've got just a couple announcements. I don't need to reiterate most of them. Homework 1 is going to be due as usual on Tuesday. Okay, good. So we'll get that done, get that submitted. The other thing that you need to bear in mind is that prior to Tuesday's lab, you'll want to submit your first pre-lab. Otherwise, today I am going to jump in because we are so lucky to have with us a guest lecturer today to talk about the human microbiome. So I'm taking only a little bit of your time today, and I want to pick up where we left off last time. We were about to talk about some of the other implications and applications of microbiology, and I was hoping that I could pick a little bit on Ella. And where are you sitting? There she is. Ella is really into baking sourdough bread, right? Tell us a little bit about that. It tastes so dang good. Oh my gosh. And what's in it? And microbes, flour and water and microbes. So the microbes are really what makes sourdough sourdough. And these microorganisms include not only the yeast that are responsible for the rising of the bread, but they also include lactic acid bacteria. Lactic acid bacteria, aka probiotics, the bacteria that are good for us and are health-giving. So who else, and I know Sean is one, brews beer, right? So tell us a little bit about the different kinds of brewing that you've done. Okay. Yeah. Have you ever tried a sour beer? Yeah. What's different about those? You don't like them, right? It's not to your taste. But the cool thing about a sour beer is that a sour beer not only has yeast, right, eukaryotic organisms, but it also has lactic acid bacteria, probiotic bacteria. So sourdough, sour beer, both have those lactic acid bacteria. Now, does anybody know historically when in ancient Egypt, when they started brewing beer? Yeah, Riley. Really good. 1500 BC. Yeah. Whoa, people have been brewing beer for a long time. We've really co-evolved with beer, which is good news, right? So we can say there are a lot of economic applications: food production, beer, wine, bread, soy sauce, yogurt, sourdough bread, pickles. All of these are fermented foods. I've gotten lately, well, you know I love some kombucha, but I've also gotten lately to like a lot of kimchi. Kimchi is a Korean fermented food, and maybe you also like a little bit of on your bratwurst. Oh, help me. Sauerkraut, right? Sauerkraut is also another probiotic. No, Riley's not on that one. Okay. So else do these lactic acid bacteria do? Well, they preserve the food as well. So those organisms that might cause food spoilage are suppressed by the lactic acid made by the lactic acid bacteria and by other components secreted by those bacteria. But let's turn to another economic application. And when we think about this one, I'm going to find Eva, if I can hunt her down. Where are you? There, she's in the back. Eva is our plant person. I don't know, Eva, where your knowledge area is, but can you tell us a bit about the roots of plants and what's going on down in what we call the rhizosphere? Yes. And nutrients, right? Do they do that alone? Okay, good, good. And they are assisted by an entire population of microorganisms. In fact, the rhizosphere is like an urban city of microbial action going on down there in the roots. And in fact, we sometimes say that plants don't have roots, they have mycorrhizae. Well, fungi, in the form of these symbiotic organisms that live in the roots of plants, are assisting in that process of bringing in nutrients and water. So there's a huge role they play there. Now, how in the world could that relate to removing oil, TNT, and other incredibly harmful chemicals from the environment? And Raleigh's got a question I'll answer while you're thinking about that, and then Patrick's going to...
Don't microorganisms contribute directly into legumes, creating nitrogen usable by other animals?
Right, perfect. So one of the specific types of microorganisms that lives in the rhizosphere is called rhizobium. And rhizobium lives symbiotically with the roots of legumes, where rhizobium actually fixes nitrogen and provides that to the plant. It's a love-love mutualism, right? That's what we call that kind of symbiosis when it's love-love, that they exchange goods, so to speak. Nitrogen fix nitrogen for organic carbon. What's your thought, Patrick? They eat it. Exactly. And in fact, the prokaryotic world is the only world that eats for dinner TNT. Who can tell us about TNT? Highly explosive. Do we want it in our environment hanging out? No. Same with oil. We don't want it in our environment hanging out. We use microorganisms to remediate it. And Eva, here's the sweet thing. Have you ever been out on the bike path out? So you start in Optimus Park and you head around the loop, right? It goes out towards the railroad. It's a pretty cool place. Yeah. And guess what? There was 10,000 gallons between like 1928 and 1982 of creosote sunk into the ground out there in that now beautiful spot. Anybody know what creosote is? Perfect. Yeah, good. It's preserving wood, railroad ties being the pivotal example in this particular case. And so now we're using what is called phytoremediation, which is utilizing the rhizosphere of the plant, of the tree, and augmenting the growth of the bacteria in that rhizosphere to eat the creosote. And I hate to say it, engineers, they tried like this dual kind of purge system, just a mechanical removal. It didn't work. But the phytoremediation is working. So this is one way in which we see an application. Here it is. This is actually a picture taken by one of my prior students. His name was David, out there on the Greenbelt, where I love to go roller ski with the ski team. So see there, they're using phytoremediation to remove that creosote. So not only are microorganisms good at bioremediation, phytoremediation. Bioremediation is the use of microbial life to remove those pollutants. Phytoremediation is when it takes the form of the symbiosis with the plant. So both are kind of a similar vein. Jake, you had a question. Yeah.
So is it the plant helping bacteria eat this stuff, or like I'm confused on that?
Yeah, no, it's the bacteria within the rhizosphere they are augmented by the fact that the plant is there, so their populations are increased, and if we can keep that relationship going, we can keep the bacterial populations high that eat it. So it's not the plant. Yeah. Yeah. Yep. Exactly. Yeah. Sort of like an enrichment, you could say. You could think of it that way, Patrick. Yeah, that's a great question. So that's a chicken and egg question to a certain extent, right? Sometimes they're inoculating the seed on the surface of the seed. They're not inside the plant per se. They're soil dwellers. Yeah. Unless you inoculated them in some way. And there are actually rhizobium inoculum. So you can actually do that. Has anybody done that in gardening where you're growing peas and beans and you actually add a rhizobium inoculum? Yeah, Scott.
On the farm, we would do that whenever we grow pinto beans or hay.
Yeah.
They're legumes.
Right.
So we inoculate the soil with planted seeds.
Right.
So then you have a much higher net.
Right, without adding.
Nitrogen.
Yeah, aka fertilizers. Right, totally rad. Okay, so some of the others of you will be interested in another economic application. That is that picture, again, that rhizosphere where we have a lot, and in any soil, a lot of microbial life. These guys are going to be vying for the space and resources, aren't they? They're going to be competitive with one another. So one of the things that they secrete to try to fight off or to dictate the species composition around them is antibiotics and also other antimicrobial compounds. Yeah.
Can bioremediation be used in the ocean or just in soil?
Yes, very good question. And it's Elena
Elena.
has a beautiful question. We can also use this in the ocean. Where have we used bioremediation effectively in the ocean? Katie, oil spills. We have, right. Now, there are some gives and takes on this, okay? There are some gives and takes on this because we don't necessarily want to change the existing species composition of the ocean when we're trying to clean up these spills. So on that note, this is beautiful because it segues perfectly, and we're not going to get to this, but you know what? I want to give Christopher extra time today. So we'll start there, and you can maybe ponder this till next time. I'm going to let him hook in, and he's going to talk to you about the ecosystem of the microbial life on us. And here's the coolest thing. This quote comes from one of the students from last semester. She said, Rachel, I can't believe how Christopher's lecture and our focus has changed the way I look at everything. Every time I see Kristen walking by, I think that's just their own little world. So
you just stole my thunder.
Yeah, isn't that beautiful? Did I ever tell you that quote? Isn't that beautiful? I loved it. So we're going to have him talk. Now let me give him a quick shout out. So Christopher is a graduate student, and there was one of you, I'm trying to think who this was, who asked, does anyone do research on microorganisms at the University of Wyoming? Here is me up close and personal, one of those people. He works in Dr. Naomi Ward's lab. She is a brilliant educator and a brilliant scientist and a phenomenal person. And Christopher is doing a project that he's going to tell you a little bit about, but he's going to also tell you more broadly about the human microbiome.
Sorry, I put you over here. But you might be wondering why I'm showing you an image of the Earth. And what I want to do is I want to put you all in the perspective and get you all in a little bit of a window into my brain and into the way that my view of the human body has changed since I've taken on this project about 2.5 years ago. And that starts with the Earth. This is Earthrise. It was taken by Apollo 8, spacefaring people, and it's the first color image of the Earth that we ever had. And it's been designated by many as the most influential photo ever taken. And what I want you to do is I want you to look at this photo. I want you to look at the surface level of the Earth. And what you can see is you can see clouds, and you can see land, you can see the oceans, right? We all know that that's the Earth. There's a lot more than what we just see on the surface level. And I think you can take this into the human arena. So when I'm out here looking at all of you, I think of it at a much deeper level than just looking at a surface. So we can start, and I can put this in perspective with a few things here. Does anyone have any idea how many cells are within the human body? 10, 14, yeah. So it estimates anywhere from 1 to 24 I've seen. We're going to say 1. So we're going to be modest. So these 1 trillion human cells are represented within this little restroom symbol as colored dots. So those are your cells. They build your tissues, your organs. They make you, right? But you aren't just you. There's a lot more going on. These cells, this gigantic circle of cells, represents the roughly 10 to 1 bacterial cells that you have. So you're outnumbered roughly 10 to 1. And that may not be entirely true, which I'll touch on in a second. But for the sake of the argument right now, we'll say 10 to 1. This is our forgotten organ. This is the entirety of the microbiome. We can look at this another way. We have this idea of our second genome. Does anyone know it's estimated how many genes are within the human genome? Drop the number. So it's anywhere from 20 to 24,000. So we'll say 20,000. That's here. That's these A, C's, T's, and G's that are within the symbol. Those are your genes. They determine whether you're tall or short, whether your hair is blonde, maybe brown, maybe your eyes are blue, maybe they're red if you're that genetically unique. They make you with your genes. But if we look at the number of bacterial genes that exert an effect on your body, the number is actually about 100 to 1. And that's pretty profound, because that means that roughly genetically speaking, you're only 1% human, and you're 99% microbe. It's pretty awesome, I think. So when we take these two ideas and we put them together, we find maybe the most profound, and in my biased opinion, because I've researched the microbiome, the coolest fact I've ever heard in my entire life. And that is, like that Earth, that's an ecosystem, you're a walking ecosystem. So with you, you carry all of your microbes, all the interactions that you have, abiotic, biotic, intricate webs, networks, all these communities of microbes that are in certain areas of your body. So everyone you've ever known, everyone you've ever met, everyone in this room is a walking ecosystem, which is pretty damn cool if you really think about that. You walk across the Earth, all of this stuff going on that at a surface level you have no idea until you take your lens and you go a little bit deeper. I think that's really awesome. So a disclaimer that I mentioned is that about a month ago, Cinder and his colleagues came out with this study that says our estimates may be wrong. It may be more that bacteria and human cells are about 1 to 1. And in fact, they have this quote, which I think is kind of funny, and it says that indeed the numbers are similar enough that each defecation event may flip them in the favor of human cells. So at some point in time, you may have more bacteria and you may have more human cells. So to segue with the disclaimer and with the way I presented this at the beginning, I want to name drop my famous science, or my favorite scientist of all time, that's Carl Sagan, and he talks about science. And importantly, within this quote, he says, in science, there are no sacred truths. That's why it's such a brilliant tool for us to use to understand things. When another idea comes out, such as Cinder and his colleagues saying that maybe it's only 1 to 1, we can't just disregard that because the prevailing belief is 10 to 1, right? Now, I still present the 10 to 1 at the beginning, not because it's more profound, not because it's cooler, but because we need this to be reproduced. We need more estimates to come out to say, yeah, it's closer to 1 to 1. And only then can we move on as a scientific community and as future scientists out here in better understanding what's going on with these numbers. And they're also estimates, so it's kind of hard. I mean, there's no grad student that's going to sit there and connect or count every single human cell and every single bacterial cell. That would be just the worst. So Morgan had asked a question, an interesting question, and that's if our bodies contain 10 more microbes, 10 times more microbes than human cells, how are we able to keep these in check? So even if they contain just 1 to 1 ratio, how does our body keep this in check? And then how do we keep them from overtaking our bodies? And we first need to introduce this idea of scale and size. So bacteria are very, very little. We have about a 2-micron bacteria represented in this small circle, diameter of a human hair with the bigger circle. And so you can see that, roughly 25 of those little circles are probably going to fit within the big circle. So we have this idea of scale and different sizes. So we can have 10 to 1 bacteria in our bodies and then not overtake us in terms of scale, but also our immune system is pretty good at fighting off infection, or when you get an infection, remembering that you have that infection and continuing to fight it off. And also our microbes are just wonderful at keeping everything in check. You have commensal microbes all over your body that make sure bad microbes aren't moving in, setting up shop, causing you infection. It's not foolproof, but it's pretty close. So, I'm going to take you on a tour through who our microbes are, where they come from, where they are at the moment, and some of the things they do. But first, we need to take a brief foray to the forest. I believe Hunter had posed a question about next-gen sequencing and what sequencing is. And the reason the microbiome is so young, it's only about a couple decades old, is because we had to transition from these culture-based methods of the early 1900s and the mid-1900s to more high-throughput approaches using computers and next-gen sequencing. 99%, I think it's estimated, of microbes can't grow in a lab, so they're really hard to study. So with the advent of technology, the advances of computer, what we're able to do is we're able to take these populations like a swab from your skin. We're able to extract all the DNA that's in that, and that includes your human DNA. And then what we can do is we can throw it into a sequencing machine that will give us a readout of what those sequences are. It'll give us quality of the sequence. It will trim out all the human-related sequences, so our results aren't being influenced by those. And then we can throw them in computer software. Namely, a big one that I use is called CHIME. It's unimportant what all of that stands for, but basically it's just a way to take these sequences, put them into a pipeline, assign them to which bacteria they belong to, in terms of phylum, in terms of genus, sometimes down to the species level, which is really cool. So you can, with this awesome technology that we're able to do, which has only emerged in the last couple decades, is go to places that we are pretty confident microbes are there, that are unculturable, and we can begin to learn a lot about them just based upon DNA sequence. So who are they? This is just a rough estimate of the composition of the human body in terms of human cells, bacteria, archaea, fungi. And as you can see, it's overwhelmed by bacteria. And this is still in the context of 10 to 1 rather than 1 to 1, so just keep that disclaimer in mind throughout the whole time. I don't want to discredit fungi or archaea. They are also important in our health, but I'm going to focus and be very bacteria-centric for this talk, just because they are in the larger population. There's a lot more we know about them. But some of the important fungi that are on you and that are more well-known are yeast, such as Saccharomyces, bread, beer, things like that. You might know penicillium. That's one of the fungi we originally got penicillin from. And then archaea, primarily methanogens, which are in your gut. That makes sense. Think about it. So who are these bacteria? So they're pretty much dominated by 4 phyla, and that's firmicutes, bacteroides, proteobacteria, and actinobacteria. And those may be words you've never heard of, because when I first got into this project, they were words I'd never heard of. So let's put a few faces with those names. So the phylum Firmicutes comprises, among many different species, of note are staph, your streps, lactobacillus, which is one of those lactic acid probiotic bacteria. Bacteroides, a little less popular. People may have no idea what these are, but that's Bacteroides, Performonas, in your mouth. And Proteobacteria, probably some more famous pathogens are within this group. We've got E. coli, Helicobacter, and Pseudomonas. You might come across, I think, E. coli and Pseudomonas in the lab, at least in non-harmful form. And then actinobacteria, which are going to be bifidobacteriums, which are also lactic acid in some cases, and probiotics, as well as micrococcus, which I think you'll come into contact to in the lab. So where do they come from? So babies are quote-unquote sterile during gestation, and I put quotes on sterile because at one point we thought urine was sterile, thought blood was sterile, mostly sterile, but not always. So it may be that babies are sterile during gestation. That's where we're at right now, but we keep finding microbes where we didn't think they were, so it seems unlikely that they'll remain sterile for all time. So the very first contact you have with microbes is going to be dependent upon mode of birth. So if you're born the traditional route, your first microbial contact will be through your mother's skin, as you might expect. If you're born with a C-section, mother's birth canal, the skin of your mother's birth canal, traditionally. If you're born C-section, your first contact is going to probably be with your skin as she holds you, right? Makes sense. Can I get a show of hands of who thinks mode of birth makes a difference? All right, who doesn't think mode of birth has a difference? A couple? All right, so it turns out that it's a really complicated answer. We're not fully sure, but it seems more and more with research emerging that it's going to be important on the microbiome, and it's been shown to be important in terms of health. I was a very unhealthy baby, and I was born by a C-section, and that's the case with a lot of people that I know. Not, again, 100%, but anyway. Does anyone have a guess as to maybe where the next contact with microbes is going to be after you're born? What was that?
Breast milk?
Yeah, breast milk. Right, this nutrient-powered, awesome drink that babies drink to make sure they're healthy, right? So it's packed with all of these good germs, all these probiotics. It's also packed with something you may or may not have heard of called a prebiotic. Basically what those are nutrients that only the microbes can use to survive, that your body's unable to process. So breast milk comes with these probiotics that begin colonizing your gut, setting up shop, and then it's also packed with these prebiotics that help feed and maintain and keep those probiotic organisms established so they can not be sick.
I have a question.
Yeah.
So does the human body produce prebiotics like naturally once you're an adult?
Yes.
Produce these bacteria?
No. So as far as, well, I mean, yes and no. So I don't think that that's fully clear. Rachel might know. As far as I know, no. You have to take those from a foreign source. That's, I believe, where we're at. So.
And thinking about the food that you ingest as a food source for your probiotics or for your microbiome. So the food you take in can be prebiotic.
Yeah. So is there a possible deficit? Like you might not be actually getting enough prebiotics?
Yes.
We'll learn about that.
We can talk about that, yes.
All right, so the next place that they're coming from is going to be the environment, from other humans, from pets, from food and water and plants and dirt and everything. And babies are dirty and gross, and they get it all on everything. So they get all of these microbes from the environment all over them. And what you have to keep in mind is that human-associated microbiomes have evolved to be associated with humans. So they're not necessarily great at surviving in the environment. And if you flip that coin the other way, environmental microbes aren't necessarily going to be great at establishing themselves on your bodies. So essentially what that means is some of the microbes you come into contact with the environment will establish themselves and some won't. The majority probably won't. So Jade had asked a question. This is really cool. This is research that came out a couple of years ago, but she had asked, how can microbes vary so much from person to person when we're always in contact with each other? Wouldn't there be more sharing of each other's microbes? And there's a lot of sharing. So you actually, everybody in this room has this microbial cloud, like this little aura of microbes that are just like hanging out in the air around you. So when you're in close contact, like you all are right now, you're sharing each other's microbes. Might kind of sound gross, but keep in mind that most of these microbes are good germs, or at least non-harmful germs. They're not out there really to make you sick. There's a lot of other, if you're in a relationship or if you have pets at home, depending on what interactions you're having, if you have a cat, you guys start adopting similar microbiomes. It's not full carryover, but it is at least some. Or if you're in a relationship, depending on what you're doing, you're around this person all the time, you can start having a convergence of your microbiomes, which is actually kind of really cool. So just keep in mind as you walk through the world that you are an ecosystem, and also the ecosystems live all around you.
Ecosystem convergence.
So where are these microbes now? This is going to be dependent on a whole host of factors. It's going to be your age, what gender you are, geography, and I put altitude up there because I did a replication study, and I'll talk a little bit about it later, and they had originally done everything in Boston, and I repeated everything here, and it seems like we're having changes, and eventually we'll have to start teasing out what these are, but as you might imagine, if you're at a higher altitude, you have lower oxygen availability, so microbiomes could differ based upon that. Microbes are very picky about their oxygen selectivity. I don't know if you guys have covered that, but if you haven't, you definitely will. As well as body weight. We'll talk a little bit about malnutrition and obesity-associated microbiomes and some really cool experiments a little later on. And then diet, which I put in red because that seems to be a very, very huge influence on your microbiomes. then we have this idea of an enterotype. So microbiomes do differ. You know, your left hand and your right hand are going to be different. The person on your left or right, as Rob Knight has said, is going to be 50% different than you. Maybe less, maybe more, things like that. Unless you're sitting there perpetually, as we talked about. So we have these enterotypes. So we have roughly 3, I believe. We have 3 types, and essentially what that means is that we can, at least to some level, start classifying an organism based upon what microbes we have. As scientists, we love to put things in boxes and categorize them and label. Not always great, but we're trying our best, and especially in microbiome research, it's really hard, but we have these 3. And essentially, they're very influenced by where you live. So yes, geography, but also generally where you live determines a lot of what you're eating and the types of foods you're eating. So with Western diets, you have this dominance of bacteroids, which means you're consuming animal proteins, amino acids, saturated fats, things like that, more traditional like American style diets. And then the other one I think that's really cool is this type 3, which it's a varied thing based upon a lot of different things. We actually can't put it in one geographical location like we can some of the others. But it's characterized by a bunch of polyunsaturated fats and alcohol content, which means that some cool, I guess, or fun research has shown that type 3 is typical of most college students while they're in school. So does anyone have a guess as to maybe which might be the most common?
Type 2?
Type 2? It's actually type 3. So again, I don't know if that says about teaching population about alcohol consumption, but here it's pretty awesome.
Well, across the world, everyone has something of alcohol in the culture that they're on.
Right. Good point.
So this is a really cool image that can just show you, based upon body location, these are the same colors as what I had presented before. And you can see that based on body location, the composition of these 4 dominant phyla, and I think they threw on a couple more, is going to vary, which is, as you might expect, you're not going to have the same microbes as in your hair as your oral cavity, so long as you're not consuming your hair, yes. But a really cool thing, so Helicobacter, I'm not going to talk about it a lot like I did last time, but it's pretty popular in the news. Every now and then we get these outbreaks of it in our stomachs, and it's really interesting to look that if you look on everybody's right, that a stomach that's positive for Helicobacter is dominated by proteobacteria, which means it's dominated by Helicobacter. So if you are a carrier or if you do contract Helicobacter, odds are your stomach is going to be just really dominated by that, which can be good and bad. Bad if it goes unchecked and you can get ulcers. Good because it can actually, in some cases, it's been shown to protect you as well, which is kind of this weird double, it's like coin thing. Sometimes it's good, sometimes it's bad. That happens a lot actually in the microbiome. So I talked about where they are, what they're doing, who they are. Now I'd like to get into a little bit more of some of the things that they're doing. And this is a really large list of things that we have linked to the microbiome, having influence in, things that we have discovered and verified and reproduced to actually be vastly influenced by the microbiome. And it's just a gigantic list of all kinds of cool things. One of the things that I think is really cool, we'll talk about this left column because of a question that Delta had asked, but it's been shown that sometimes in the gut we can have bacteria that are modulating our neurotransmitters in our brain, so they're actually affecting our behavior on a somewhat observable level. So they send their signals through the vagus nerve, which goes up to the brain and tells the brain maybe, I'd really like to eat that candy bar because I'm a bacteria that really loves those sugars. And so they can actually start to influence like things that we're doing and choices we make. Still in its early days of that research, so there's still links, not direct roles, but we're getting there. And things are getting funded at a better level now than they were a few months ago. So I think that's a really cool one. This whole left side has to do with essentially keeping you healthy and helping your immune system stay healthy. So Delta had asked a 2-part question, the first part of which was if we get sick and recover, do our microbes adapt to help us prevent another sickness like that one? And the answer is essentially yes. And also, they help our immune system adapt. So most of the time, your immune system is what's going to be responsible for making sure you don't get sick again. But in fact, your microbes can actually help train your immune system to maybe recognize a pathogen. So when you're young, the microbiome will really help tell your immune system this is good, this is bad, I'm allowed to be here, this isn't allowed to be here. It helps your immune system develop as a result of this. There's a lot of crosstalk between the two, especially within the gut where you have a lot of neurons and a very important immune system to your health. There's a ridiculous amount of crosstalk between these. So much so that Lactobacillus, a probiotic, has been shown to actually upregulate things such as mucin to help protect your mucosal barrier in your stomach. Meaning, when it's colonizing that barrier, pathogens aren't able to come in and colonize it, cause inflammation, and make you sick. The second part of your question has to do with microbes playing a role as to why people in certain cultures live longer than others. Environment excluded, geography excluded. And this is a little less clear-cut. We don't quite know the answer to this question. What we do know is that diet does seem to affect how long you can live. It's not foolproof, but some indications suggest if you have a healthy diet, you can live a longer, better life, right? And then it's not too much of a stretch to think that if the microbiome is so influenced by your diet, that potentially the microbiome in the diet and maybe taking prebiotics and probiotics could vastly affect not only your health, which we know, but maybe potentially prolonging your life. So I've got a little bit of ways to go on that. A lot of this research, even though we've come very far in the amount of time, in the short amount of time that we've been studying the microbiome, we have a ridiculously large black hole of information that we don't know. We're getting there. So when things fall apart, so when there's disruptions in your microbiome, perturbations, outbreaks of things, maybe you ate the Chipotle that had E. coli in it or whatever, is something in the news, all types of things can go wrong with your microbes. And as you can imagine, all of these influences that affect your microbiome, there's a lot of stuff going on. So it's not really a defined set of things. So this is basically, again, a comprehensive list. It's probably somewhat overwhelming of things that we've made links and things that we know the microbiome directly plays a role in. But unfortunately, in microbiome research, we have this issue of what we do most of the time is we take descriptive approaches. So we take a sample from a diseased patient and a healthy patient, or a diseased patient that went into relapse, that's now healthy. And we just basically describe what microbes are there. And that's great, that's the first step, but it's only an association. You can only make an association. Well, we observed this microbe in the person while they were diseased, but not this group of microbes. So they associate those with the diseases, and we have this hurdle in microbiome research where we have to transition from association to causation. And the way we do that is both gross, but also absolutely beautiful, in my opinion, but also is different. And so a really great example of this is how we discovered a link with obesity, malnutrition, and our diet and our health and the microbiome. Basically what these researchers did is they had normal mice, and what they were able to do is they were able to collect feces, poop, from people that were obese, and poop from malnourished people. And what they did was they fed them to these normal mice. And in the case of the obesity-associated microbes, the mice got really, really fat, right? So the fecal transfer, I guess I should mention, is basically with mice, they shove a tube down their throat after the poop's homogenized and they coat their fur and those microbes establish. It's really gross, but as you can imagine, we can't do that with human beings. Well, I mean, we can. We'll talk about that in a second. Anyway,
Yeah.
Skinny mice, normal mice, got fat, and then they took the malnourished species and they gave it to normal mice, and they got skinny. And then they could even do the reverse, where they take their fat mouse and they transplant in the malnourished microbes, and they start to return back to a normal mouse, which is actually really cool. And so fecal transfers become this emerging, awesome thing. And they are disgusting. They also are really, really awesome in terms of human health. And if any of you have ever heard of C. diff or Clostridium difficile infection, it seems to be that in some cases, not all, that a fecal transfer can actually save these people's lives, and save their colons from, and their intestines from just getting inflamed with an incurable antibiotic-resistant superbug. Right? There's also been links, and again, I'm not going to touch on all of these. If you guys have any questions, I'm more of an expert on some than others, but after, just contact me or email me or whatever. So there's been links between cancer in the microbiome and changes in the microbiome. And at this point in time, they're mostly still links. There's a lot of cancer research obviously going on. But a few things we do know is that certain genera are associated with tumor growth. So some of these probiotic organisms can actually suppress tumor growth, which is really cool because we may have a way to at least suppress the growth of the tumor in the hopes that maybe there's a tandem treatment that we can throw in that will actually get rid of the tumor or potentially just surgery to cut it out if that's a feasible option. There's also not direct evidence, but at least some research has suggested that there's a relationship between microbes that cause inflammation. So if you have microbes that are causing a lot of inflammation in the colon, sometimes that can lead to cancer due to just destroying your insides to a level where your body can't cope. You start accumulating mutations and develop cancer there. Those are still just links at this point. Inflammatory bowel diseases. So these, I guess I hesitate to say, are close to my heart. Obviously, I don't like these, but this is where my research comes in. So we work in this ballpark of a field. Specifically, we work on Hirschsprung's disease and Hirschsprung's associated enterocolitis. But in the grander context of inflammatory bowel diseases, a lot of really cool stuff is coming out, linking microbiome disruptions in the microbiome to the cause of these diseases. And unfortunately, for me as a researcher, it's not quite so clear-cut because there's a lot of factors that go into causing these diseases. There's environmental factors, there's genetic factors, there's dietary factors, there's microbiome factors. So it seems to be that it's not just going to be the microbiome being disrupted causes this, but a whole host of things together, which makes our job difficult, but also really, really fun because we can start making connections between these and start designing treatments based upon what we're finding. So Crohn's disease is probably the most, well, it is the most prevalent, probably one of the most known inflammatory bowel diseases. And again, the cause of this has genetic and environment, dietary, and potentially microbial input to it. So a little bit on my research, I guess. I didn't really talk about this last time, but so we found in 2012 that there's evidence for a disrupted microbiome and causing this Hirschsprung's associated enterocolitis, which is essentially just an inflammation of the intestines as a result of having had Hirschsprung's disease, which is essentially babies are born with a non-functioning latter half of their colon, so they can't poop. So a lot of things can, a lot of symptoms and terrible things can come out of that. There's surgeries to correct it, which is nice, but you can still develop this enterocolitis, which is terrible because these children can die from it. So we found that there's this evidence for this disrupted microbiome, and right now I have experiments currently ongoing to do fecal transfers to see if we can take disease-associated feces, transfer it to a non-disease mouse, and induce colitis to see if potentially it's only the microbiome that's causing it. We've done these on a base level, and it doesn't seem quite that that's going to be that easy, but we're still hoping, and we're going to test a bunch of other stuff as well. So anyway. So biotic warfare in the future. So where we are at with current treatments in terms of like C. diff and things like that are basically fecal transfers, which again are really cool, but also gross. So we're still very much in the early days of this. And at first they were shoving tubes down people's throat and pumping feces into their stomachs to establish these microbes, which as you can imagine is really, really gross. And it also, complications from having a feeding tube down your throat is you potentially just vomit up all that stuff that they just put in your stomach, which is also very gross. So we've transitioned to pill forms, like frozen fecal samples encapsulated in a pill, which unfortunately you have to make the pill acid resistant, so they have to be clear. So they still look like this, which is still gross, but much better alternative to having the feeding tube shut down the throat. And these have proven just as effective as the oral lavage. So while gross, still very cool. And I mean, if you had the choice between dying or taking this pill, I'm pretty confident I'd be taking the pill. So, a little bit more into the future are potentially in tandem with fecal transfers, or maybe on their own, we can take probiotic organisms and prebiotics and put them in some combination to start maybe ameliorating these diseases and helping cure them. That's an idea that I have potentially for treating the disease that I work with, is in the combination of a fecal transfer, maybe they need prebiotics to help establish themselves and things like that. And I think we're going to see, at least to some level, a decline in antibiotic usage. We have this like serious rise in antibiotics prescribed, and to correlate with that, we have this serious rise in superbugs that are antibiotic resistant and terrible, and we can't treat them, and they're killing people. And so that's due to a whole host of things, but if we can start designing treatments that are prebiotic and probiotic based, we might be able to start fighting these superbugs. And so I think as we see this potential decline in antibiotic usage and all of these things that we're surrounded with constantly that are meant to sanitize all of these bad things that are on you, they're actually sanitizing all the good things you have too, which is not very good for your health when you think about it. So we may actually start having non-hand sanitizer that maybe unsanitizes your hands, things that are actually good and can help prevent infection, rather than wiping the plate clean and allowing something that's bad for you to come in inside your shell. Hannah had asked a really cool question, and this is my attempt to get you guys involved in my favorite research project. It's not the only one out there. I should say that there's Bowel and collective gut or something like that. There's a bunch of different ones. This is my favorite one. I've met these people. They're wonderful. They're nice. They're awesome researchers. And it's called the American Gut Project. And so basically for $100, and there's several levels of packages, you can either do a fecal, a mouth, or a skin sample, and you can, they'll mail you a package with all the steps to collect these samples, and then follow their steps, mail them all their stuff back, and then they'll sequence it. They'll give you, I think if you pay like $50 extra, they'll give you this awesome poster that says, these are what microbes you have, these are how they compare to the rest of the world, or the rest of America. There's a British gut project, there's all kinds of gut projects. This one is super awesome because it's just, it's $100. Like, come on. I mean, that's not that expensive. I guess as college students it might be, but ask for it for Christmas.
Yeah.
It's really awesome. And it not only is it cool to know what's there, it helps our field understand on a big crowd-funded level what microbes everybody has rather than just the select few that they're saying. So, I'll skip this. This is how you can contact me. I don't have office hours this semester because I'm not TAing, but this is my office. This is my lab down here. Email, probably best. I'm not always the best at getting that, but it's a good way to contact me with questions. Or there's my phone number. That's a risky thing to give you all, but I don't care. Text or call anytime. I am so excited about science that it doesn't bother me.
Well, let's just put the version on the
Okay, we need to
Save up. That's what I've been doing. So great.
Have a fantastic weekend, everyone. Oh, by the way, feel free to get back to now and start answering each other's questions.
Be interested in my story.
Did you have a?
I
do the healthcare? Yeah. Sorry, I didn't put that in.
No, you're totally good.
It was just really long.
I just
Yeah, don't forget about the
I
dating
wanted
you
talked about work with some like Crohn's and that kind of stuff. I wanted to take a
Yeah, let me just hit something real quick. Yeah. Just 1 second. Give me 1 second.